Good afternoon. My name is Dr. Clement Muka. Today, we're gonna engage in a HIV chat. And the reason for this HIV chat, I got a motivation from a patient who was very bitter, who said to me, "Doctor, I was born HIV positive. My mother died when I was 15 years of age. I'm 27 years of age right now. And I was told that I was born prematurely." She says to me, "I, rootless stamped, son before my time, I am determined to prove a villain and hate the idle pleasures of these days." You know, I was like defeated. I met this patient at a rehab. I'm a rehab doctor. And I decided, look, we're going to have a chat about this HIV. Let's have a chat. HIV, human immunity deficiency syndrome. HIV belongs to a group of viruses known as lentiviruses. Lentiviruses because they go against the central dogma of molecular genetics that says DNA, RNA, protein. It goes against that. These are retroviruses. So this particular one, HIV-1, because we categorize HIV into HIV-1 and HIV-2. In other words, HIV-1 and HIV-2. HIV-2, we don't get it much. We don't get it at all. Actually, it's not the one that causes the AIDS, this acquired immunity deficiency syndrome. No. It's HIV-1 or HIV-1 that causes an acquired immunity deficiency, what we call AIDS. HIV-2, you get it in places like Senegal, that part of Africa. And it's not as knocking as HIV-1. Well, people manage it that side. Well, I don't have experience of that, but I do have experience of HIV-1 because that's the one I treat on a daily basis. I say this, it belongs to a group known as lentiviruses. And which other lentiviruses do you have in mind? I have, for instance, the simian immunity deficiency virus. You know, this one is different from HIV-1. HIV-1 targets non-dividing cells, whereas the SIMF or the simian immunity deficiency virus targets the dividing cells. So the non-dividing cells that we are talking about, we're talking about the CD4 T lymphocyte. The CD4 T lymphocyte, and it targets a receptor there, that is the CD4 T lymphocyte receptor. But it has got co-receptors The co-receptors are the CCR5 and CXCR4. And these are the virus. You know, it's so nice to talk about this because in a place, I don't know, Corsica, they don't get HIV-1. They don't get HIV infection at all. They don't get HIV infection at all. Because the CCR5 receptor has changed to become CCR5 delta 32. CCR5 delta 32 is not an entry point for HIV. Entry point for HIV is CCR5 delta, is CCR5. CCR5, that's the entry point. It enters using that. So that core receptor is a bad core receptor. It's not helping us. It's actually, that's the one that kills us. And CXCR4. So these viruses, what is responsible for the entry? The HIV virus it's an enveloped virus, number one. When you say enveloped virus, from the envelope, you do see receptors sticking out. The GP120, for instance. And lower down, you have the GP41. The GP120 is actually, you know, HIV is referred to as it's referred to as a virus that goes to a particular receptor. It's a tropic virus. It goes towards that particular receptor. It's a tropic virus. So what is responsible for the tropism is the GP120. When you say GP120, what's that? GP120, glycoprotein 120. It's a glycoprotein. It has got, on the envelope, it has got these glycoproteins which are sticking out. So when you see those from the envelope, you see the GP120 sticking out, you know that's a tropic. That's a tropic. That's what's responsible for the tropism of HIV. It will gravitate towards a certain receptor, which is CCR5 or CXCR4. It will gravitate towards that. So it doesn't fumble, it doesn't go anywhere, like fumble and hope to bump against a receptor. No, it goes straight to that receptor. So it's a tropic virus. HIV is a tropic virus. That's what we need to take home. It's a tropic virus. So when it gets in there, so it goes to this receptor. So when it goes to this receptor, the core receptor, that is CCR5. Now, the virus is in contact with the target cell, that is the CD4 T lymphocyte. But then, the virus and the receptor, they are not yet combined. They need to, the plasma of the virus, the viral membrane has got to be in touch with the plasma membrane of the target cell. To do that, it needs to be facilitated to do that. That's when it uses the GP41. It's for contact, the actual contact between the two membranes. The plasma membrane, this is of the target cell, and the viral membrane, that is the HIV, combine together. When they combine together, then it gets a chance of depositing. The work of GP120 is finished now. The work of GP41 is finished now. Then now it deposits into the cytoplasm of the cell. It deposits. the viral capsid. That's number one. The viral capsid is the one that contains the viral genome, is the one that contains all the genes, the viral genes. The viral genes we're talking about, we've got genes for the GAG gene, the Pol gene, the ENV gene. The ENV gene is for the envelope, to be able to make that envelope with all those GP120s and GP41 sticking out. So there's a gene for that, a gene for GAG. GAG is the glycoprotein that's gonna form, that's gonna cover. It's gonna cover the virus when it's actually formed. So what you have, what you are taking in, you are taking in GAG gene, you are taking in the Pol gene, you're taking in the ENV gene. The ENV gene, that is ENV gene, EMV, envelope gene, ENV, the envelope gene. So those are the 3 genes you are taking in. Why are you taking in these genes? You are taking in all the genes. In fact, that's not all. Those are the 3 genes I'm mentioning. That is the GAG gene, the Pol gene, and the ENV gene. There are other genes which need to be taken in, which are in this capsid, viral capsid. You've got other genes like, they're genes for accessory proteins. Genes like the TET gene, the NAV gene, that is the NAF gene, the REF gene, the VIM gene, you know, the TET gene. So all those genes, those are accessory. Those are the genes which make sure that infection is a possibility. In other words, the HIV doesn't fail to infect the CD4 T lymphocyte. It doesn't fail. So you've got accessory genes which are going to be they are going to be transcribed and translated to become proteins. And as accessory proteins, they are going to help the virus to actually infect the target cell. To infect the target cell. To infect us. So what type of genes do you have? You have the GAG, you have the Pol, you have the ENV, and then you have the accessory genes like the TET, REF, NEF. You've got VP, VPI. You know the VPI? You've got VPU. So VIF is the last one. VIF is the last one. Wow. You will say it's not necessary to know this, but you must see a situation when we think we've gone against the infection, and then this viral protein just comes in to make sure that HIV is successful in actually infecting us. So these accessory genes are so very much necessary. And what do you have as the main one? The actual RNA genome. The actual RNA genome. But remember, HIV is an RNA virus. It enters as an RNA. That's why, and it has in it enzymes which are responsible for converting this RNA to become DNA. Remember, it has got to be DNA in order to be integrated to another cellular DNA. It can be it can be an RNA that's integrated to a DNA. No, that is why in it this viral capsid, that's why it has got reverse transcriptase. It has got integrase enzyme. Those are functional in actually making sure that this whole process is a success. The first one is the one that's responsible for naming these viruses retroviruses, because it's a reverse transcriptase. It's the one that reverses RNA to become DNA, viral DNA. It has to be viral DNA so that it can get integrated. Remember, HIV, like all viruses, it's an obligate parasite. It depends on the cellular, on the target cell, because the target cell is capable of replicating, making copies of itself. But the viral genes which are entering, they are not capable. The viral genes which are entering, which I was saying, the gag, the pol, the end, they are not capable of transcription and then translation to become proteins. Remember, that's exactly what should take place. Inside the nucleus, there has to be transcription. There has to be translation is done in the cytoplasm. So it will go out having what is known as an mRNA, messenger RNA, the one that has got the message now. This is how you are going to make the proteins. But it's going to be made by the ribosomes in the cytoplasm. So, but then the entry into it's transcription. You want those mRNAs, so that you should be able to tell the ribosome, this is the information of making a protein. And that's what you need. So after this being dropped into the cytosol or cytoplasm, in other words, this viral capsid being dropped there, it's not only carrying those genes that I was talking about and all those genomes, RNA, that's not the only function. The other function is to move from where it was dropped to the nucleus. So trafficking becomes important. So trafficking to the nuclear pore, it must go to the nucleus. And it must get inside the nucleus. So it's so strategic. The way it is made, they call it, the viral capsid is made in such a way. It's a cone structure. It's made of the P24 proteins. The P24 proteins. They form the lattice, so that it should be able to get inside the nuclear pore. It should be able to get inside the nuclear pore. And when it gets inside the nuclear pore, it gets into the nucleus, then it starts uncoating. It uncoats now, so that transcription should be a reality, or transcription should be a success. Because if it was to get into the nucleus and sit there and closes itself like that, all uncovered, it has got to uncoat so as to expose these genes which are to be transcribed, so that when they are transcribed, they form the mRNAs. And those mRNAs, they go out now to the cytoplasm. For what? For translation. Translation into what? Translation into proteins. because that's exactly what you wanted. You wanted the formation of protons, viral protons, but you couldn't, because the virus is an obligate parasite. It is unable. It is unable to make copies of itself. It can't make copies of itself because it needs a what? It needs an mRNA, a messenger RNA, which has got the information for making protons, viral protons. But those viral protons it can now go out of the nucleus, go to the cytoplasm, to the ribosomes of the cytoplasm, where translation is going to be done. Translation into changing now what we read here to become we are going to make the protein according to this template, which is from the nucleus. That is your messenger RNA. This is good if you know that. So basically, that's what you need to do. When it gets out of the nucleus, then the gag, remember we said there's a gag gene. That gag gene, when it gets out there, it has already been transcribed. Now it's going to be translated to become a protein. It forms now what is known as a gag polyprotein. A gag polyprotein. That gag polyprotein is so clumsy, and elongated and all that. It has got to be cleaved. It has got to be cut. It has got to be pruned. And what prunes it is a protease enzyme. The protease enzyme is, you see, when the gag polyprotein is formed, and you are having all even the accessory proteins, you're having all the polygins and all that, and polar. Now you're having them as proteins. But there's a problem. The virion, or the virus particle that has been formed outside because of these proteins, viral proteins, it is very sluggish and unable to infect another target cell. Now it has got to be made to be infectious. How do we make it? By pruning it, cutting, cleavage, cutting, to make sure that it becomes an infectious virion, or infectious HIV particle now, a virion, an infectious one. And what is responsible for it is the protease enzyme. So there were 3 enzymes in this viral capsid. The ones which were responsible in the nucleus was what? Was the reverse transcription, the reverse transcriptase. It was responsible for reverse transcription. It's the one that made a new virus. In other words, made a double-stranded virus, a double-stranded DNA. DNA is always double-stranded. So the RNA was changed and it was made to be double-stranded. Some people call that a provirus, a proviral DNA. That's the DNA, that's the viral DNA. That's the one that has got to be integrated onto a cellular. It has got to be integrated onto a cellular DNA. The cellular DNA has got all the capabilities of replicating, so it doesn't have a problem. So if this one joins, it's also going to be replicated. All the, like whatever has been to form a new, an mRNA, the viral mRNAs, so that all those proteins that I was talking about, they should actually come to be, proteins and not just, in other words, the translation of their mRNAs is going to give rise to proteins, viral proteins, and those viral proteins are the ones which need to be cleaved by what? By the protease enzyme. So the protease enzyme works after the integration has been done by the integrase enzyme in the nucleus, integrating onto a cellular DNA. You know, we are using now powerful drugs like dolutegravir, which inhibits that integration of the virus, that integration of the proviral DNA to integrate it onto a cellular DNA, right? So it inhibits that. So when we are treating these patients now, we're using what is known as TILD. In other words, tenofovir. We're using lamivudine, and we're using dolutegravir. Dolutegravir goes against the integration, right? So these are what we need to sort of. These TB chats will keep on talking about this. Just, doing these TB chats just to make sure that we understand what's going on, and now there's a drug that goes against that very viral capsid. That very, it's an injectable. I don't know, people are not talking about it, but it's coming. It goes against that. It goes against that container of all the genes. It contains all the genes that are to be transcribed, all of them, including the viral genome, which is the main one, accessory genes, the GAG, the POL, and the ENV. It goes against that. It goes against the container of the viral capsid. And that's what we'll be talking about next time. Thank you very much, guys.